Hey everybody, and welcome to Unit 5, Lesson 2, Exploration 2. And today we're going to be talking about mitosis and cytokinesis. And really this is the process of the DNA dividing and then also the cell dividing into two equal cells. So as we scroll down, this is what we went over in lesson 1, but this is the cell cycle. And here we have interphase. Interphase is going to be made up of G1, S phase, and G2. Remember in G1, this is where, let me scroll down, this is where the growth, and we copy the organelles. In S phase, and this is very important, this is where the DNA replicates. The DNA, and remember replicate just means to copy. So we are going to have one set of DNA here. However, during this S phase, every single chromosome is going to copy, and we're going to have two of them. And then we're going to get some additional growth going on in G2. So we have growth in the organelles copy. Here the DNA copies. So after this spot right here, we have twice as much stuff in the cell that when we started with. And now we're going to grow again because in this blue portion, this is mitosis and cytokinesis. This is where that DNA in the cell are going to split in half, and these are going to separate from one another. So as we scroll down, the definition of mitosis is by which the cell divides the nucleus and its contents. And cytokinesis is the process by which the cell cytoplasm divides, but it's the actual cell and the membrane is cytokinesis. So as you scroll down, we've got a little video here that the book provides of a cell going in. It's going to prepare and then copies, right? That's mitosis. That's what we're going for. And now we have two identical cells. That happened real quick, so I want to watch it one more time. The cell is going to grow a little bit. Cell's going to grow there, divides in half, and now it's going to go separate ways. It completely divides, and now we have two cells that are absolutely identical. And that's how we got all of these cells. So we scroll down, here's a picture of the cell, and this is what happens in prophase, kind of before we get ready to divide. Centrioles are going to produce these products called spindle fibers, and I want you to think of like tug of war, right? So what's going to happen is these spindle fibers are going to attach to the chromosomes, and that's what's actually going to pull the chromatids apart from one another. So as we scroll down, it talks about the cell cycle in detail, and here's what happens. So when we get into interphase, as we know, the DNA is going to copy, and the cell is going to grow. In mitosis, it's going to be made up of 4 parts, and this is very important that you know these 4 parts. They are prophase, metaphase, anaphase, and telophase. And a lot of people give that an acronym of PMAT, P-M-A-T, and that gives you the order as well. So in prophase, what's going to happen is the chromosomes are going to condense and the nuclear envelope is going to go away. So we're going to lose the nucleus, and now the chromosomes are going to have the entire cell to work with. And then the centrioles are going to do what's called move to the poles. So they're going to start moving opposite one another, just like we think of the North Pole and South Pole being opposite on our planet. So these are going to move opposite. And see here, the centrioles are going to be opposite. In metaphase, the spindle fibers are going to attach, and all of these chromosomes are going to line up right down the middle. And you can notice, we call this the equator, right down the middle. And you can notice that the chromatids are ready to be pulled apart by these centrioles and these spindle fibers. As we move into anaphase, we still have the centrioles on the outside, but as you can see, they're starting to pull. These spindle fibers are starting to pull these chromosomes towards this centriole, and these are getting pulled towards this centriole. And as you can tell, we have a green one going this way and a green one going this way. Pink and pink, blue and blue, white and white. And what the colors mean, and chromosomes are not colors inside of our cells, but what these colors are trying to signify is that these are the same chromosome. And so we have one copy going to this cell and one copy going to this cell, or what will turn into those cells. And then as you can see in telophase, we now are going to have the nuclear envelope come back. But now we have 2 nuclei. So these chromosomes that went this way are now these. These chromosomes that came this way are now these. And we're starting to get a nuclear membrane around it. The centrioles are going to start to move away. The spindle fibers are going to start to disappear because now we have 2 nuclei. And then cytokinesis is the process of the cell membrane and the actual cell dividing in 2. So this is why in cytokinesis we actually have 2 pictures. But what you can notice is we started with this cell, And the goal of the cell cycle is to get 2 of these. And as you can see, this cell right here is exactly the same as this one, which is exactly the same as this one. These are all the same cell, exactly the same. As we scroll down, here's a video that's going to walk you through the process. So here we're in prophase. The nuclear envelope disappears. You can see that our centrioles are moving to the poles. Now we go to metaphase because the chromosomes are going to line up in the middle and the spindle fibers attach. Then we're going to go to anaphase. You can see these are going to start to pull apart. Right here is anaphase. And now we're going to go into telophase where the nuclear envelope, here it comes, starts to form around it. And then we're going to get cytokinesis right there where it comes apart. And now we have 2 exact copies of that cell. As we scroll down, I want to talk about one thing, and it's talked about asexual reproduction. And what we're talking about is organisms that are going to reproduce without going through sexual reproduction. And the main process that we're going to use is binary fission. And binary fission is asexual reproduction, meaning without sex, in which a cell divides into 2 equal parts. And here's how this is going to happen. And here's kind of an example on the side. First of all, the DNA in prokaryotic cells are not in the form of chromosomes. Are not going to condense. They're going to stay in the form of chromatin, which is going to be very stringy and almost like soup. Okay, and what's going to happen is it's going to attach to this end of the cell, and this version is going to attach to this end of the cell, and then the cell is just going to start to elongate. And as it does that, it's going to pull these 2 sets of chromosomes apart, and then the cytokinesis is going to get to split down the middle. So binary fission is going to happen a little bit different than a eukaryotic cell. Here's just a quick little diagram that I want to go ahead and fill in. So DNA is replicated. Well, we're going to need both copies. Anytime we make a new cell, we're going to need 2 copies. So that's going to happen in both binary fission and mitosis. DNA is highly coiled. That's in mitosis. In binary fission, in prokaryotes, the DNA is not going to highly condense and coil. DNA attaches to the cell membrane. That's going to happen in binary fission. DNA is often circular. That also has to do with bacteria and prokaryotes. 2 identical cells are produced. That is both. That is very important because that's the whole goal. The whole reason we do this is to get 2 cells that are absolutely identical. And then the DNA attaches to spindle fibers. Remember, those spindle fibers are what pull the chromosomes apart in mitosis. So once again, that is the process of mitosis. I'm going to scroll back up and do a quick review over the steps. The steps are by far the most important part for our class here. And here it is again. Remember, we're going to have interphase, but then we're going to get into mitosis, and that's prophase, metaphase, anaphase, telophase. And here we're going to get ready to divide. Chromosomes line up in the center with the spindle fibers attaching. In anaphase, they pull apart. And then in telophase, the nucleus come back, and then in cytokinesis, the cell membrane actually breaks apart. So there is Unit 5, Lesson 2, Exploration 2, and can't wait to see you guys in class.